Good morning, I'm Bruce Korf. I serve as Chief Genomics Officer at UAB and was asked to be the kind of first half of a two-part lecture today on sort of precision, personalized genomic medicine. I think you'll hear from Jim Cimino in the second hour, and I'll focus really on the area of genomic medicine. I think the link that I sent with the slides didn't work, and we only learned that first thing this morning, so that's why you don't have a printout. So I'll take credit for having saved you from wasting some paper. And I think the slides will be made available to you later on. So I want to start with this famous but obviously old picture, and it gets to the question of what do we mean when we say personalized medicine, precision medicine, and I'll get into genomic medicine. And a lot of people use the term personalized medicine, but they find it a bit objectionable because, well, for one thing, maybe it implies the existence of impersonalized medicine, as if anybody really practices that. Well, arguably many do, but they also feel like personalized medicine has been the way medicine always has worked. And I guess I think this picture really kind of hammers home what is, in a way, the difference between personalized and precision medicine and why the latter term now tends to be preferred. The physician in the foreground here, I think by anybody's definition, is practicing personalized medicine, meaning that he is absolutely raptly attentive to the problem presented by his patient, this obviously sick child. But the other point here, personalized though it may be, is you can tell from the distraught expression on the parents that this physician has absolutely no idea what to do to help his patient. And so it's very personalized, but it's absolutely imprecise. And in my mind, that's really what the modern sort of approach to medical care offers, is to use knowledge, and that knowledge includes many different facets, as you'll see in a minute, to offer not just personalized care, but precise care, to understand the pathophysiology of a person's medical issues, and to offer a form of care that really answers to the individual needs of that person. And if you think about individuality, what makes each of us different, or put differently, why we're not all identical twins, there are many things that feed into that. One of them is ancestry, and that's what I'll focus on in a certain sense today. By ancestry here, I'm referring to genetics and the composition of an individual from a genetic perspective. But by no means should that be viewed as the entire equation here. There are individual matters of environment, the homes we live in, the cities that we live in, and so on, which influence our health. There are cultural issues that influence health, how people perceive what we might call good health even. And finally, personal experiences are different for each of us, how we would view the possible options, for example, that might be presented to manage a health problem. And the hope is that as we learn more about what constitutes individuality, we'll be able to customize care and define an individual risk of disease, not the average for the population, but one person's risk of disease, to predict their response to treatment and to avoid side effects, so as to customize the treatment. Some people say that precision medicine amounts to giving the right drug to the right person at the right dose at the right time, and I think that's fine as far as it goes, but I don't think drugs are the only component of what constitutes care, so I'm not personally that fond of that way of thinking about it. So what I'll try to do in this next few minutes is to focus on just one facet, the first one I mentioned, that of ancestry, and translate that into genomics, and take a look at the spectrum of rare disorders to common disorders to cancer, some words about epigenetics, and then finish with a summary of some of the programs that are happening here at UAB, which hopefully will be of interest to you as things that you might get involved in. So rare disease genomics is not a new concept, and this is maybe the first example of a genomic disorder that was discovered in 1959. You'll recognize the phenotype of Down syndrome in the child shown at the lower right. This is a photo of chromosomes as they appeared around that time in the early 1960s, where you could separate them and order them by size and the position of the centromere. And what was known as of 1959 is that instead of the 46 chromosomes that most of us have, children or anybody with Down syndrome will have 47, and this smallest group of chromosomes included an extra chromosome, which at the time was arbitrarily defined as chromosome 21, trisomy 21, a genomic disorder because it involves multiple genes and produces a distinctive phenotype. So we're talking, many decades of experience. This is cytogenetics as of the relatively recent past, and this picture is meant to depict a child with autism spectrum disorder where this particular technology has made a big difference. The technology of the day is a cytogenomic microarray. You take DNA from a person that you're interested in studying, you take a reference sample, you mix them together, and you let them compete to find their homologous sequence on literally a chip, a glass slide, that contains hundreds of thousands of chunks of DNA from different parts of the genome. And if the two, which are differentially colored, let's imagine your patient's DNA is colored in green, literally by binding it to fluorescent markers, and the reference in red. And basically what you find is that if they are equal in terms of the representation of a given sequence, they fall along the midline. But way down here you can see that there's some area where your patient's DNA didn't compete effectively with the reference, indicating a very tiny deletion that might just be a few thousand base pairs that would have been invisible with the light microscope, but nevertheless can delete multiple genes, or the opposite could occur where they duplicate multiple genes, and this leads to a phenotype. And in fact, the pickup rate for copy number changes with this technology, which we use every day now, has gone from about 5% in individuals with intellectual disability and autism spectrum disorder to pushing around 20% now. So it's not 100%, but the pickup rate is vastly improved. And what that means is diagnoses can be offered to individuals who previously were mysteries. So the genomic era in what you might describe as single gene disorders dates back to about the 1980s. And at that point in time, as gene mapping technologies were developed and then as gene cloning approaches were added, the ability to identify genes based on their location in the genome, which has come to be called positional cloning, in some ways an obsolete term now, but historically interesting because it really made a big difference in the way we think about genetic disorders. Up until then, if you wanted to identify a gene that was associated with a particular phenotype, you had to know what the protein was that was involved. So for example, sickle cell, which would be the first example of a human genetic disorder that was attributed to a particular gene, we knew that, or not we, but they knew that the red cell was the source of the phenotype, and globin was the major protein in the red cell. And you could show by protein electrophoresis that globin wasn't structurally correct. And ultimately, by actually sort of backing into the genetic code, which was only just being elucidated in the 1950s, the gene variant was identified. But for so many other things, we had no idea what the underlying pathophysiology was. This shows an example of cystic fibrosis, I imagine a familiar disorder to most of you. Up until about 1987, nobody had any idea what the underlying gene was. The gene, however, then was mapped using genetic markers that could be shown to segregate in families together with the phenotype. Then a cloning exercise was done where chunks of DNA from what turned out to be chromosome 7 were mapped in relation to these markers, and ultimately a gene was found that was consistently mutated in individuals with cystic fibrosis. Turns out to be a chloride channel. Knowing that, as you'll see later, has not only enabled diagnosis, but even a customized approach to treatment. The sad truth, think of this as clinical investigators, is that if you took all of the literature on the genetics or pathophysiology of cystic fibrosis and put it in a library room and locked the room, and this was before the days of electronic journals, the world would be no worse off because all that literature really was obsolete as of the time when the gene was identified. Not clinical things maybe, but any of the genetic studies were rendered irrelevant once a gene which was previously not known to exist was found. And there are lots of examples of the same thing where we had no idea what the gene was, but we knew something about its location in the genome because of gene mapping, and it ultimately led to finding the gene. And so the trajectory of gene discovery over the last 15 or so years has really been exponential. But what really has fueled it and brought it to a new level is the idea of next generation sequencing. This is a graph maintained by the National Human Genome Research Institute, which has a pretty good idea of what it costs to sequence a genome because they are paying genome centers to do that, and they know how many genomes they're getting back for the money they put in. So in 2001, it cost about $100 million to get a genome sequenced. And you can imagine the challenge of getting an insurance company to cover that if you were trying to do it for an individual patient. So only really a very small handful had their genome sequenced at that time. And if you track and realize it's a logarithmic y-axis, from 2001 to 2007, there was a tenfold reduction of cost, but still $10 million is a pretty hard argument. And notice too that it was tracking with Moore's law, which you'll recall is the statement about the density of transistors on a computer chip doubling about every 18 months. It's why the computer in your pocket is better than the things that used to fill a room 30 years ago. And geneticists were kind of proud of that. But then you see too that the price plummeted around 2007, and it's tracked more or less to the present day. And it's, as you can see, it kind of bounces around a little, but it's touching the x-axis, which starts at $1,000. This price point is one where you actually can have a conversation about insurance coverage, and we do, and we get it done now increasingly. By the way, it's not such a great thing that we have exceeded Moore's law, because what you need to analyze the data are computers, and they have turned out in many ways to be rate limiting, although there's been a lot of progress there as well. So what made this possible is the technology generally referred to as massively parallel sequencing, and there are many technologies in use, and I don't think it's that important really to know the details, but let me walk you through a kind of oversimplified idea just to give you a flavor for it so it doesn't seem like magic. And the chemistry and the approach might not be the same in particular techniques that you might use, and it's going to be a moving target. But fundamental idea is you break the genome up into 100 base fragments, short segments. Of course, in a 3 billion base pair genome, there are going to be a lot of these 100 base fragments. You adhere them to a solid substrate, essentially think of it as a glass chip, and then you use an enzyme to amplify the DNA. So instead of one strand at any given point, there's a cluster of identical strands. So you get more of a signal. A good metaphor is bristles on a brush. Think of the brush standing or sitting on a tabletop and the bristles sort of up in the air. You then use each of these as a template for a new DNA synthesis reaction, but you color code the 4 bases. In this case, A is red, T is yellow, and so on. And you allow this, by manipulating the chemistry, to just build the new DNA strand one base at a time. So in other words, you let the first base bind to wherever it's going to bind by DNA complementarity, you freeze it, and then picture this brush with a camera in the ceiling looking down on it. It takes a snapshot. So what happens is these dots correspond to the location of these DNA strands, these bristles, if you will. And this one here, the first base that bound was an A, you could tell because it's red. And the one next to it, which is this, the first base that bound was a C because it's green. That picture taken, you allow the next base to bind. This one here, which the first one was an A, now it's blue, so that must be a G. And this one up here, which was blue the first time, now is yellow, that must be a T. Well, you only have to do this 100 times. And then by reading the successive patterns of color on these photos, you can piece together what the 100 base sequences were. But what you've done is you've sequenced the entire genome, albeit in 100 base fragments, in a matter of hours instead of in a matter of years, which is what it was before. And because we know the kind of generic human genome sequence as we have for the last 15 years or so, a computer can piece together these 100 base fragments and place them where they belong in the genome, and then scrutinize them for any variant that deviates from the reference genome. And that fundamentally is how it's done so quickly and so cheaply. The first success in identifying a gene associated with a human genetic rare disorder was in this very obscure disorder called Miller syndrome, dysmorphic facial appearance, abnormal limbs, as you can see, and intellectual disability, but it was vanishingly rare and had never been mapped because there were too few families to be able to do the mapping. But it's interesting to see what they actually did. They had 3 families with 4 affected individuals. Kindred 1 had 2 children, A and B, who were affected. And then there were Kindred 2 who had 1 child, and Kindred 3 had another. So we're talking 4 affected persons in 3 families. And they sequenced them all. We'll go into a little more detail about what they really sequenced in a second, but just for the moment. And in just this one child, A, there were something like 4,670 variants that were heterozygous, meaning there was only one of the 2 copies of the gene had that variant. And there were 2,863 where both copies of a particular gene had a variant. They then said, well, this is a rare disorder, and this is now 2010, so things have changed a lot, but there was even then a database of common variants, because enough people had been sequenced. They had some idea of variants that were frequent in the population. They said, this is not going to be a common variant. And so they found when they used one database just for the dominance, there were only 641 of those 4,670 that were not in one particular database and 898 not in another. Then they said how many were not in either one. The answer was 456. When they looked at the biallelic, both alleles were affected. There were only 31 in this child that were not in either database. And then when they looked at the sibling, for a recessive, there were 33. For a dominant, there were 400 and some odd. Then they used a computer algorithm to look at what these variants were and make an estimate of whether they were likely to affect the function of the protein or not. And to make a long story short, in the recessive category, there were only 6 that were rare variants that were shown to be, or at least predicted to be, damaging computationally in this first child, 12 in the second child. And then they asked, between these 2 kids, did they share any? And the answer is yes, only 1. And then they looked at these other 2 children from other families, and they found only this same one was not present in common databases in either of those 2. The computer algorithms didn't entirely faithfully predict that they were damaging. But to make a long story short, this turned out to be the gene, just eliminated as not a common variant and shared by multiple affected individuals. What they actually did was so-called exome sequencing. Remember, the genes are actually divided into exons, which are the coding sequence, and introns, which are intervening sequences. The introns are spliced out and the exons knitted together to make a messenger RNA. Something like 1.5%, maybe even less, of the genome consists of exons. It's a little bit daunting to realize that everything you learned about molecular biology, probably in college, maybe in high school, only really was true for 1.5% of the genome, but that's the way it is. But most of the variants that seem to be associated with disease reside in those areas. And so what now can be done is take genomic DNA, break it into fragments, and there are libraries of chunks of DNA that are known to correspond with exons. You use them to bind to the same corresponding chunks in the genomic DNA. You throw away the rest, and you just sequence that. So you can sequence about 30 or so million base pairs of DNA and find most of the gene mutations that occur in at least medically relevant issues. I will say that exome sequencing is probably on its way out. The technology to sequence the entire 3 billion base pairs now is pretty well established and pretty efficient, and you actually get better coverage of all of the exons from a whole genome than you do from an exome. Right now, if you were to send a clinical sequence, it would probably still be an exome. I don't think that's likely to stay the case for that much longer, but that's how it is today. How many variants do you actually see in a genome? The actual total number of variants might be in the range of 4 or 5 million in a whole genome sequence. What was done in this table was to look at so-called novel variants that hadn't been found in databases of common variants, broken down into missense, nonsense, synonymous changes that don't change the amino acid and so forth. And you see that there are many hundreds. And interestingly, you notice in the US population a larger number in African Americans and a somewhat smaller number in European Americans. This has to do with the sort of evolutionary history of populations. Since humans really had their origin in Africa, the migrations that resulted in European populations, or for that matter others, were small subsets of a much larger population with less genetic variation because they represented the founders of whatever those populations were. So you often see a larger number of variants in individuals of sort of recent African ancestry. We're all of African ancestry eventually, but more recent African ancestry, usually the numbers of variants becomes larger. How do you find a gene? Now back to that Miller syndrome, if you will. One way is to take multiple families, look at all the variants, and say if they have a phenotype in common, what's the intersection of these? And that it doesn't prove it's the gene, but it does provide a candidate gene. So that's one way of doing it. Another works if you've got a big family, mainly with a dominant, as shown here, and you look for segregation. There's a variant you find in this person similar to the variant you find in other people who are affected. And if you have enough people in the family, there shouldn't be any variants really besides the one responsible for the disorder that faithfully are transmitted through the family to all the affected individuals. The third way is to look for instances in a so-called trio of a child and both parents. And when we can, clinically, that's what we do. One thing you can look for is, assuming both parents are unaffected and the child is affected, if it's dominant, that's probably a new mutation. So you look for a variant present in the child, not represented in the parent, and either parent. And new mutation is not as rare as you might think. Every time a sperm cell or an egg cell is made, there are about 30 mutations, which is a kind of daunting thing. You want to really be daunted. If you take a population of germ cells, like about 100 million, which is about what you might find in even a population of sperm, essentially every nucleotide is mutated in one or another sperm, or to put it in slightly different terms, every genetic disorder ever described or that ever will be described is represented in some sperm cell in any man, and the same could be said for egg cells in a female. Of course, most of the mutations are not medically relevant. They exist in parts of the genome that are not expressed, but some of them turn out to be, and that's how children wind up with so-called de novo genetic variants. And this has been a very powerful way of doing sequencing for diagnostic purposes. And finally, you can look at outliers, compare individuals at two extremes of a phenotype and look for gene variants you find in this group that you don't find in this group. So this is happening every day here. This is an example of a child we diagnosed in the UAB Undiagnosed Diseases program by genome sequencing. The child had a neonatal encephalopathy, seizures, intellectual disability, severe neurologic dysfunction. This is his brain MRI, a big cyst around the area of the cerebellum and brainstem hypoplasia. Nobody else in the family had this. We found two different variants that were both de novo, this SP10-1, heterozygous variant in the child, which turned out to be a duplication that would cause an out-of-frame transcript and has been shown before to be pathogenic. And then another variant, which was a single base change and a splicing variant that would affect RNA processing. Neither parent carried either one of these. We're pretty sure the SP10-1 was the driver. This is a MRI from a published case with the same variant, and it shows, again, the cerebellar hypoplasia, though not as profound, and also brainstem hypoplasia. There were enough cases that we felt the match for our patient was pretty good. It's not that rare to find two mutations that might be pathogenic in the same person. You would have thought not, right? We tend to use the sort of Occam's razor, try to find the simplest explanation. And you would normally assume a person will have one or another genetic disorder, not multiple. But that's turning out not always to be true. And that's important because when you define the phenotype and you look for a gene variant that describes that phenotype, you might never find one because the phenotype might be the intersection of two different genetic conditions. And that happens more and more now. Quick word about a UAB resource. We have an undiagnosed diseases program here. The staff are shown in the slide here. I won't have time to go through everybody's role. We see children and adults with essentially chronic undiagnosed, sort of mysterious conditions. The one I showed you a minute ago was an example of that. But we have multidisciplinary evaluations. We do whole genome sequencing together with HudsonAlpha, which I'll introduce later. And we actually have monthly meetings where we get together and talk about our unsolved cases with consultants from all over the kind of community representing pediatric or adult specialists. And we also have monthly meetings where we get together on WebEx with the HudsonAlpha group to talk about variants that have been identified and try to decide if we think they're relevant or not. And both of these are really, I think, fascinating meetings, and we're certainly open to having other people attend. We've had, during the last 4 years, something like 400 referrals. We've accepted just under 300 for detailed review. We've actually accepted for evaluation 188. What that means is the balance were referred to other clinics because it was felt that they might have had a kind of a single system disorder better addressed there. Completed 100 evaluations, and so far have 57 that we've diagnosed that didn't have diagnoses before. And it represents a wide variety of medical problems. Genetic diagnoses might be the biggest chunk of the pie, but you notice there are many other kinds of diagnoses. Not everybody's diagnosed by genome sequencing, although that is the one source of, completely unexpected diagnoses. And I guess it's worth saying that we either diagnose things that, like in that first case I just showed you, we would never have thought of in a million years. I'm sure that diagnosis would never have otherwise been made. Occasionally we find things that in retrospect we kind of wish we had thought of. And then we also find things that we're not quite sure what they mean. When you're interpreting genomic data from a clinical perspective, or trying to attribute it to a phenotype, the spectrum you might see is from pathogenic variants to benign variants, and we all have millions, as I told you, of variants that are not medically relevant, or they are phenotypically relevant, like brown eyes versus blue eyes, but not medically relevant. There are things that we're not so sure about, that we think are probably pathogenic, or we think are benign, but you can't be 100% sure. And then this kind of vast midground of variants of unknown significance, which means what it says, that we aren't sure. whether that variant is or isn't pathogenic. And trying to get to an answer there is another real opportunity. We find lots of these as we sequence individuals in the program, where we find a variant, it's real, we know it's there, but we're not able to prove that it's caused the phenotype. And this represents, I think, a great opportunity for collaboration with basic scientists, because many of these will turn out to be phenotypically relevant, but it will take additional work to prove that. I'm not going to go into details. This is a pretty complicated slide, but I just want you to know, and the reference is given here in Genetics and Medicine, there is now a formal set of criteria to decide if something is pathogenic or benign or somewhere in between, with different levels of evidence based on many different factors, including does it segregate with the disease if it's a dominant, is it a de novo mutation if it's a new sporadic case, have other cases been found, is there some knowledge of pathophysiology, many different things go into it. There are now databases that are maintained, and this is showing what's called matchmaker exchange with a lot of different databases where if you find a variant and it's never been seen before, you can check and see if somebody might have noticed it in a case that they've been working on. We use Gene Matcher a lot. We put in the variant and the phenotype, and it matches and says, has anybody else put that same variant or variant in that same gene with the phenotype? And they actually link you together. You can exchange emails then, and then with the family's permission, exchange information about the phenotypes. And from that, sometimes an N of one becomes an N of many. And there are many examples now of gene discoveries that have been catalyzed by Gene Matcher. There is, just so you know, a standard nomenclature used, the Human Phenotype Ontology, which is now a way of being sure that you describe a phenotype in a consistent way. And it not only matters in terms of telling a person the name of the condition, but sometimes you can begin to do work that results in treatment. This is one example, not because of genome sequencing, but it is an example of a targeted treatment. Tuberous sclerosis, a dominant genetic disorder that affects many systems, but prominently the nervous system. Either of two genes is mutated, which is a regulator of mTOR, which is a protein involved in control of cell growth. When these genes are, either of them is mutated, you get hyperactive mTOR. And mTOR stands for mammalian target of rapamycin, which was originally described in yeast and then found to be a mammalian cell. And it was a kind of convenient thing because it was called target of rapamycin. Rapamycin is an antibiotic which tends to modulate this protein, and since that's how it was discovered, that's what they called it. I suspect it will be the only time we will have the name of the treatment printed on the name of the gene. And rapamycin turned out indeed to shrink tumors, including the subependymal giant cell tumors and renal angiomyolipomas. Subsequently, a similar rapamycin-like drug was developed called Everolimus, which is now FDA approved for treatment of these lesions. I don't send children with this lesion to neurosurgery anymore. We put them on medication. And that wouldn't have happened if we didn't know what the genes were. Another example in cystic fibrosis, which, as I mentioned earlier, is a chloride channel. There's been effort either to find ways to prevent degradation of the mutated gene or to potentiate the effects of the chloride channel in the presence of the mutation. The first FDA-approved drug, ivacaftor, this is pulmonary function on treatment versus off. It only works if you have a particular mutation, which is not a common one in CF, about 2 or 3%, I think, of the population at most. But if you're one of those people, it's really a dramatic improvement in care. And now they've developed yet another drug, and they're gradually chipping away at this mutation by mutation. All right, so that's rare disease. Now let me just say a few words about common disorders, things like asthma, hypertension, and diabetes. The idea is there's a genetic liability at birth, and that by itself doesn't give you the condition. It makes you vulnerable to it. And the question is, can we identify the genetic factors so we can modify their effect on the environment? Very often, this is approached using single nucleotide polymorphism association studies, hypothesizing a particular allele. We'll call this allele 1. And here, where there's a C instead of an A, is allele 2. And you do a case control study in this hypothetical example, asthma versus non-asthma. You see allele 2 present in 30% of people with asthma, only 10% of those not. And so that statistically can be shown to establish that allele 2 is associated with an increased risk. That does not mean it causes asthma. It might mean that it's just located near another gene variant, which is associated with asthma, but the association at least is established. One of the first examples of the genome-wide approach, not taking a candidate gene that you think in advance might be related, but just scanning the whole genome for any variant that might be related, was discovering this complement factor H polymorphism in age-related macular degeneration. This is the signal of association for gene variants across the genome. It tends to be called the Manhattan plot because it's like looking at a skyline, and what you're looking for is a skyscraper. One was found. It showed this complement factor H was associated with age-related macular degeneration. Nobody had any idea that that would have been the case prior to that. And there are lots of associations now established by genome-wide association studies. This is a graph maintained by a group in the UK. Every color corresponds with a different disorder, and every locus shows an association. So there's a very large number of these. Sometimes the associations are really powerful, like in the ApoE polymorphism for Alzheimer's disease. Sometimes they're fairly weak and not necessarily that useful clinically, although they may shed light on underlying pathophysiology. There is a phenomenon, though, to be aware of called missing heritability, which is that if you sum all of the genetic contribution across the markers that have been found associated with any one disease, it usually doesn't amount for all the calculated heritability. So if you take a hypothetical disorder where about 50% of the variation in the disorder is attributed to genes, GWAS might have only accounted for 15%, in this area here is so far unaccounted for. And that's common with virtually all of the disorders. Now maybe these heritability estimates are inflated. Maybe there are some rare variants that are just not easy to test for, or just undetected variants that are in this kind of vast undescribed area. You probably know that there have been some direct-to-consumer companies, 23andMe most probably prominently, that began to offer genomic variant analysis. They no longer report common disease risk as they did when I did it years ago now. I found I was at average risk for every common condition you can think of, and I thought that was okay. But it also has to be taken with a big grain of salt, realizing that they're really only looking at a relatively small component of heritability. And for that matter, the data sets may or may not even apply to my individual ancestry. One group did a study a few years back where they sent samples to 2 different companies, 5 individuals, and the arrows point the same direction. They got the same risk, but in some cases, like here, the arrows pointed in opposite directions. One company said they were at increased risk. One said that they were at decreased. They cancel out. It means it was garbage, really. And it turns out it wasn't because of errors that one or another was making. It was because of the particular way they were calculating risk, which was proprietary and not necessarily valid. The FDA clamped down on this eventually. You no longer get common disease scores through these direct-to-consumer companies, but a lot of refinement now has taken place, and we are looking at polygenic risk scores on a more clinical basis now as a way to predict risk of common disease. Very quickly say a word about cancer genetics and where that fits into the picture. Remember, cancer is fundamentally a genetic disease. There are instances where it's actually literally familial, where the risk is transmitted from generation to generation. One of the best known examples would be hereditary breast and ovarian cancer. But even if that's not the case for a given individual, the process of oncogenesis is one of progressive accumulation of mutations. And the more that are accumulated, the more abnormal the cell is, including the more sloppy it is in replicating its genome. Therefore, more mutations arise. There's a strong selection for mutations that favor replication and ability to grow in areas the cells normally wouldn't, which accounts for the growth of the tumor and metastasis. And the question now is, can you characterize those variants and then customized treatment by identifying targets that are unique to the spectrum of the variants in that particular person. So you can sequence normal and tumor DNA, or increasingly just tumor DNA, compare the two or look for known variants associated with cancer, produce a kind of image of what the genomic landscape of that cancer is, and then choose treatments that are targeted to particular kinds of variants. A great paradigm, and you see billboards nowadays that says our cancer center sequences your genome. The part they don't always point out is they don't always have drugs for every target that you identify. And secondly, the cancers are constantly evolving, and sometimes the variants of today that you found when you sequence are not the same as the ones that are currently comprising the metastasis. So it helps a good proportion of people, but there's still a lot of work to be done. There's also now an interest in circulating tumor DNA. When tumor cells die, they release their DNA into the bloodstream, and you can detect that, sequence it, and sometimes identify gene variants in the blood that are actually being shed from the tumor, and can use that either as an early diagnostic marker or as a way of tracking response to therapy. All right, so we have a few minutes left. Let me do a couple more things. I want to just spend a few minutes to mention some words about epigenetics, which you hear a fair bit about. Everything we've talked about so far are changes in the genome that are transmitted from a cell generation to the next generation, or from a person to another person in the population, that is to say, from a parent to a child. Epigenetics comprises heritable changes, but only heritable from one cell generation to another, and not from one actual generation of parent to child. And these are not associated with base sequence changes. What they tend to be associated with, picture a hypothetical gene with promoters, which are shown by the arrows. And what you tend to find is methylation, which is depicted here, in the promoter region. And when that happens, it turns those promoters off. So in a simple way to think about it, methylation, mainly of cytosine to 5-methylcytosine, if present at the promoter region of a gene, silences that gene. But where does 5-methylcytosine come from? So imagine, if you will, a DNA sequence where the red is 5-methylcytosine, and it's going to bind like cytosine does to guanine, which is yellow. And picture a promoter where you've methylated all the cytosines in what are called CpG islands, cytosine, guanine, and so on. And these do exist in promoters. That silences that gene. But how does the next generation of the cell know where 5-methylcytosine goes? Because there is no 5-methylcytosine sitting in the cell to be incorporated into the DNA. In fact, when the DNA replicates, the two sister strands separate, and wherever there was a 5-methylcytosine, a guanine is going to be put in, C to G. And wherever there was a guanine, a normal cytosine gets put in. So it's no longer methylated here, but there are methylases, enzymes, that will methylate DNA. And if they see a CpG, the P is the phosphate, by the way, in DNA, cluster, where it sees methyl on one strand of the DNA, which was the parent strand, it methylates the cytosine on the other strand. And that preserves the methylation from cell generation to generation. But when the germ cells are made, all those methylation marks are erased, and you end up with a clean slate.
So, Dr. Clark, so you're saying through silencing, it's correcting the...
Well, it's not really correcting anything. The silencing is in a given cell. If it doesn't need a given gene, it methylates that DNA and the gene is turned off.
Okay.
And from one cell division to the next, that's preserved. But you don't want to turn that gene off permanently in all cells. So when sperm or egg cells are made, all those methylation marks are erased, so that in the fertilized egg, those genes are all active again. And as the cell differentiates, it can be modified once again.
Okay, thanks.
Very quickly, it's not just the DNA that's modified. The histones, the proteins that bind to chromatin, are modified. We'll go into depth about all the different modifications, but these are the proteins that bind DNA and either cause it to be rendered inactive by condensing it or allow it to open up to be accessible for transcription. And there are these transcription complexes, which are actually what read the methylation marks and the histone marks and actually cause the chromatin to be compacted and inaccessible for transcription or opened up and accessible. And if you look at methylation marks, which is what the blue and yellow is meant to show, in different gene regions, that's what the blocks here are, in different tissues, you see differences. And the point here is that there's no human epigenome. There's no pattern of methylation that constitutes a person. There is a neuron epigenome and a oligodendrocyte epigenome and a pancreatic beta cell epigenome and a gastric cell epigenome and so forth. These are all distinct and different. And so you need to specify the tissue of interest. One other area, just to quickly mention, is the genetics of drug metabolism, so-called pharmacogenetics, where if you think of what happens when a drug is administered, gets absorbed, metabolized, which either means activated or inactivated, and eventually excreted, and all of these processes are under genetic control. There are phase 1 reactions, which are mostly oxidation reactions, and phase 2 reactions, which are conjugations of the drug, usually to render it water soluble so it can be excreted. And there are variants in the genes that carry out these reactions. It's a bit of nomenclature. We refer to haplotypes when we have a particular form of a gene, such as A1, on the same chromosome with another gene called B1. And then in pharmacogenetics, we often refer to diplotypes, which is having, for example, a haplotype of A1B1 together with a haplotype of A2B2. And these have been characterized in particular drug combinations, and then they use what's called a star system, where a particular diplotype, such as having A1B1 on one chromosome, A2B2 set of variants on another, would constitute a so-called star 1 diplotype, and that defines a particular genotype that might predict drug response. And here are some examples. CYP2D6 is a cytochrome oxidase. It's involved in oxidation of many different drugs, and you end up with individuals who are either rapid or poor metabolizers. It explains why some individuals with a standard dose of drug have side effects that other people don't, or conversely, why a particular dose of drug doesn't help a given individual because they're metabolizing it faster than they can actually use it. And there are many drugs that are metabolized just by CYP2D6. Warfarin, commonly used anticoagulant. There's a variant in the target of warfarin, VCORC1, that determines its sensitivity to the drug, and another variant in CYP2C9, which is involved in the destruction of warfarin so it can be excreted. And particular polymorphisms will determine the ideal dose of warfarin for a given individual and predict whether they're likely to get side effects of treatment, which is hemorrhage, or be inadequately treated. Clopidogrel, a common antiplatelet drug, is an inactive drug in the form you administer it until an enzyme, CYP2C19, acts on it to activate it. So if you have a variant in CYP2C19, you give clopidogrel and it does nothing. And the FDA actually has a warning now that you should consider testing, because otherwise you can give this drug to somebody who's at risk of clotting, and they will indeed develop a clot because the clopidogrel never gets activated. This is the warning. This is an interesting one. There's a particular HLA variant that predicts Stevens-Johnson syndrome, and it's a common variant in Southeast Asia. Common enough that in some countries now, everybody gets genotyped. They carry a card that says, if I show up and need carbamazepine for seizures, don't use that drug because I'll get Stevens-Johnson syndrome, or at least have a high risk of getting it. And it's made a huge difference. It was once quite common in some of these areas to see that devastating skin reaction to a drug, and now it's much rarer because there's universal testing. It doesn't work so well in this part of the world because there's no one HLA type that covers most people. You should know about the pharmacogenetics knowledge base, which is an online area to find information about drug and pharmacogenetic interactions. And there's a CPIC consortium that you see here, which has issued guidelines for which pharmacogenetic variants are important in given situations. So lastly, and I have 6 minutes, so I think we'll make it on time. Just a few words about resources here at UAB that you should be aware of. Heflin Center for Genomic Sciences is a university-wide center, has a genomics core lab. They can do next generation sequencing. They don't do whole genomes, but they do exomes. They can do RNA sequencing. ChIP-seq is a way of looking at areas of or proteins that bind to chromatin. David Crossman there provides bioinformatics support. HudsonAlpha, that I'm not sure how many of you are familiar with, but is an independent biotechnology facility in Huntsville, hour and a half north of here. We have a Center for Genomic Medicine together with HudsonAlpha that includes resources for research, education, and clinical care. They have really massive genomic capabilities, including whole genome sequencing there. This is a newsletter. It's hudsonalpha.org, CGM newsletters. And we will have a symposium, I think it's May 23rd, up there, and everybody's invited to participate in that. Once a year, we give a immersion course, we call it, in genomics and clinical research. This was a curriculum a year or so ago. In the fall, we do a kind of a general genomics course. You can see many of the talks are really pretty basic. It's not something that you're going to attend and then write a successful grant proposal by itself, but the hope here is that you'll become familiar with some of the things that are possible and some of the people you can collaborate to actually make them happen. Then in the spring, David Crossman runs a bioinformatics week-long course. As you see, these are both Monday through Thursday or Friday from about 8:30 to 12:30, and there he actually creates real data sets that people can get experience in manipulating. We have a T32 in genomic medicine. It's a 2-year training program for MDs, PhDs, or MD PhDs, and actually any other terminal degree actually. A 2-year program that includes some didactics, participation in some seminars, as well as in things like genomics clinics or genomics laboratory activities. And we actually have 2 slots per year, so we're very eager to see those be filled. And we have a program that I guess we'll find out about the funding, but we're optimistic about, to create a summer program for undergraduates from mostly minority institutions in the region that, if funded, will start next summer. You should know that we are heavily involved in a genomics project in the state called the Alabama Genomic Health Initiative. It's funded by the state legislature. We've had $2 million a year for the last 2, and we're hoping it extends over 5 years. We'll be doing genotype analysis in 10,000 individuals, as well as whole genome sequencing in about 1,000. We have working groups in bioethics, data management and bioinformatics, education, genomics, and participant and provider engagement with the community outreach program. The so-called population cohort, which is mainly this 10,000, get genotyping on a microarray that detects 650,000 single nucleotide variants, and we return results of actionable medical variants. And then another cohort of people with phenotypes get whole genome sequencing. The genes that we are returning results are shown here. There are tumor predisposition genes, connective tissue disorders that can cause sudden death due to aortic dissection, cardiac disorders, mostly cardiomyopathy and arrhythmia, a few metabolic disorders, and malignant hyperthermia. The principle is these are traits you might not know you have, but which, if you did know, you can do something about them to avoid a devastating outcome. We've recruited about 1,700 individuals. This is showing ethnicity. It doesn't project that well. Our participants are shown in plus signs, but you can't really see them. But we've actually had a pretty good representation of diverse populations in those that have been recruited so far. The ones in red are returns of results. I think the one that I like to tell the story of is this MYH7 is a cardiomyopathy gene. The genetic counselor, once we had clinical confirmation of this result, called him and said that you would be at risk of severe cardiomyopathy. And his answer to the genetic counselor was, that was true of my first heart. He had had a heart transplant, and he had no idea why, or the risk to other members of his family. And so this is explained. We've found other people with cancer predisposition, quite a few with early onset hypercholesterolemia. They generally knew they had hypercholesterolemia. They generally didn't know why, and they didn't know about the risks to other family members. And finally, you may have heard about it this weekend because it got a lot of publicity. We are part of the All of Us program. It's a national component of the Precision Medicine Initiative, and we are the hub of a consortium that includes groups throughout Alabama, as well as University of Mississippi and both Tulane and LSU in New Orleans. The national goal is to recruit 1 million individuals, to track their electronic health records, to have them fill out questionnaires about health-related issues. There'll be fitness trackers and wearable devices and things of that kind, and they will eventually have their genome sequenced. And the goal is to take all of these data about their sort of lifestyle to their health and to integrate it with various kinds of omics markers, genomics, probably also transcriptomics and glycomics and metabolomics and so forth, to piece together the predictors of disease and response to treatment, and through that to really sort of reap the benefits of the various both genomic and other omic technologies and informatics. And with that, I'm sorry, I'm 1 minute over, but I will cede the floor to Jim Smino.
I want to take questions. I'm happy to.
If there are, yes.
What do you recommend is the best way to deal with variants of unknown significance? We have, I'm in pediatric neurology, and we get microarrays and whole exome a lot, and they're 5 years old, and there are a lot of variants. And is there some way to like go back and look at them to see if we?
Yeah. So if the first point is specific to your question, if you have a variant of unknown significance that was identified a few years ago, it's really worth querying the lab to see if there's new information about that. That happened actually, just to give you a quick vignette. We saw somebody in our undiagnosed disease program who was labeled as having spinocerebellar ataxia. And it puzzled me because she didn't have ataxia or anything else, and how that happened. Turned out 7 years ago, somebody had sent a panel which included that, and it came back as a variant of unknown significance. They misinterpreted it and thought it was pathogenic, which it wasn't. Furthermore, when we found the report, we called the lab and asked if they had new information. Yeah, they did. It had been reclassified as a benign variant in the interim. So number one thing is to check if it's an older result to see if there's new information. Otherwise, though, I mean, for microarrays, there's a limited amount you can do. That if it's been called to VUS, it's already been probably compared with known databases. For single gene variants, we are trying to build a kind of pipeline for doing functional genomics to see if we can, for example, knock the gene out in zebrafish and replicate a phenotype as one kind of thing you could, in principle, do. So there is the possibility of doing additional functional studies, but not a simple and routine thing.
So microarrays, we don't really need to worry about. It's more of the whole exome or whole genome.
Well, mostly, the only thing I would say is that re-querying the lab to see if there's new information is still worthwhile, because some of the VUSs 5 years ago, there may have been more instances of that same variant found in the interim that might shed light on its pathogenicity. And it's not practical for the labs to chase every patient they've ever seen, because they have no way to know where they are even. So that's why, as a clinician, if you're seeing somebody and you have a VUS reported before and you want to know if there's new information, it's worth asking the lab if they have any.
Bruce, if I can ask a follow-on question. So, tumor biopsy report will typically have 300 variants of unknown significance. So do you just say to the lab, can you rerun this report? Or, I mean, you can't ask them about all 300 individually, or can you?
Well, I think there it becomes a question of, what's the data on the drivers? I mean, the reality is, if it's a cancer panel on somebody with cancer, I would bet that in the time course of while you're treating them, you're not going to have that much new information. And, the lab could always be requeried in terms of, which are the variants for which there are potential targets that you could look at. And, the data sets are constantly being updated, probably not on a week-by-week basis, but, probably more on a year-by-year basis. But, this whole area is a moving target because in cancer, number one, knowing which variants are meaningful is going to change as more data are accumulated. And secondly, having a target is great if you have some ammunition to hit the target. And right now, we have a limited armamentarium, and over time, it'll probably be a lot more complete. Okay, thank you.
Thank you.
Do people need a break? How do you typically do this? Because I can just speak at whatever speed I need to fill the available time.
Right?
No? Okay. Okay. All right. Well, all right. So my topic is health informatics, and I think before I go into what health informatics is, I need to sort of set the stage by broadly defining other informatics in general and other informatics things in particular. Health informatics, kind of the blind man and the elephant model. If you are involved in any particular area in health informatics, you tend to think that's what it is, and you may not kind of get the forward spectrum of what's going on. So I'll hopefully give you a bit of a broad range that maybe you'll find a piece that applies to your areas of interest. So what's informatics? So to me, informatics is about representation of concepts and ways that computers can reason about them. Okay, so it's not just data, it's not just computer programs, it's not just user interface design, it's not just electronic health records, it's not analyzing sequences, genetic sequences, but it's actually having a deeper representation that the computer can reason about, help you with your tasks. In order to do that representation, it's important to anticipate how the information is going to be used. It's very difficult to take something about the world and model it at the level of first principles and expect that you're going to have something that works. But and I'll give you some examples of where you might reduce that. And so you have to prioritize what you represent. And there's a lot of art to it. There's a lot of people with expertise who say, this is the part that's going to be worth modeling to do a lot of different things, and this part probably not as useful. All right, so I'm going to take an example from culinary informatics. So this is the interactive part. So I want a show of hands, one answer, pick one of these and raise your hand. Which of these is not a fruit? How many say watermelon, not a fruit? 2. How many people say strawberry, not a fruit? How many people say almond, not a fruit? How many say tomato, not a fruit? Okay, so we've got 1. So, okay, so the correct answer is 3. Almond is not a fruit. The others are all fruits. There are different kinds of fruits which might be confusing, maybe causing some confusion. Now, here's a question. Which of these should not be put in a fruit salad? How many would not put watermelon in a fruit salad? How many would not put a cherry? You wouldn't put watermelon in a fruit salad? How many would not put cherry in a fruit salad? How many would not put almonds in a fruit salad? How many would not put tomatoes in a fruit salad? Okay, so a tomato is a fruit, but it typically wouldn't be put in a fruit salad. And so knowledge is knowing that a tomato is a fruit, and wisdom is knowing that that you shouldn't put it in a fruit salad. All right, so how would we represent this in a way that a computer could generate a menu or a recipe for a fruit salad that would be careful not to exclude, without just simply listing here are all the things you can put in there, but we say let's generally represent fruits and seeds and other things that could go in fruit salads, and then we could write a program that generates a random recipe. Which is the way they seem to do it sometimes Okay, so watermelon is a berry, which is maybe why you didn't think of it as a fruit. It's a berry, has flavor sweet. Strawberry is an aril, which is not a berry because the seeds are on the outside. Also sweet. An almond is a seed. It's sort of sweet. Let's not argue over that. A tomato, a tomato is a berry also, but its flavor is savory. All right, now a berry is a fruit, has flavor. Or aril is a fruit. Sweet is a flavor, savory is a flavor, and a fruit salad has fruits and seeds, in my definition, and has flavor sweet. So you can see if I represent this knowledge in some formal way, you could write a computer program that could then decide through inferencing which of these things could go in there. It could say, oh, well, I can put sweet fruits and sweet seeds, and an aril's a fruit, and a strawberry is an aril, so therefore it's a fruit, and it has flavor sweet, so it can put that in there. Tomato, not sweet, so I wouldn't put it in there. All right, so that's culinary informatics. If we wanted to write a program to do that, that might be one way to approach it. So there's lots of informatics terms. If you Google culinary informatics, you'll actually find it occurs there something like 144 times, many of which are probably copies of my slides that mention it. But banking informatics, legal informatics, things that you would expect to use information systems haven't really latched on to the word informatics. Bioinformatics actually the big winner. But biomedical informatics, clinical informatics, health informatics, it's really, informatics really kind of belongs in the biomedical/health domain when you see it. All right, so let's take an example of a patient record, which is really what health is about, and look at some of the things that are in there. So a nurse reports a patient's glucose is 50, which is low. A physician orders some glucose and then repeats the glucose. So I use the word glucose 3 times in there, and a human, at least a clinician, wouldn't even notice that I might be ambiguous in what I was saying. They would know, so you did a blood test and you found the result was low, so then you ordered a medication and then you repeated the blood test. And so a clinician would not be confused by that. First, glucose is a medication. Well, so this was another interactive thing, but the idea is that it's a test to measure physiologic parameter, and then there's also a medication that has that same chemical that is the physiologic parameter. A computer would be confused by this because the computer would just see the word glucose 3 times and really wouldn't know what to make of that. All right, so we could represent it more formally, and we've got these 3 concepts, and we have the context that it's used in, and then we have controlled terminologies. So we can say if you're ordering something, you got to pick from this list. And if it's a lab test, here are the lab tests, some of which have the word glucose in them, but and some of them are called blood sugar or some other name. And then over here we have medications, also controlled terminology. Then knowing the context, the computer actually can help us figure out what to do from there. So we might represent now if we look at this representational scheme, serum glucose, a physiologic parameter, located in the bloodstream, has glucose, and a value is a number. And then intravenous glucose is a medication. It also has ingredient glucose. So you notice the word glucose is repeated, but in a different context, and then has a strength. And then a serum glucose test is a laboratory test, has specimen serum, and it measures glucose. So all of those things relate to the concept glucose, which is a chemical. None of those are the concept itself. They're all using that in their definition somewhere. Now a computer could start to reason about things like this. So I could write a rule that says if a physiologic parameter is low, then administer a medication that's associated with the associated chemical of that physiologic parameter and administer it into the location where it's low. And so you could actually say, and then order a lab test to measure that associated chemical. So now I've just solved the computers in medicine problem. Just basically run a test, and then whatever is wrong, give some medication to make it right, and then you're all set. And then order a test to make sure it's okay. So very simplistic, and actually we don't practice medicine quite that way. But you could see you could do some interesting things with representing how you would decide when to use a test, not by writing an individual rule for every possible test and every possible medication, but a more general rule because you've represented these concepts at an abstract level that you could reason about. So it gets a little tough, though, when you start looking at other things. Lab tests are pretty easy because somebody invented them, and there's a laboratory that does them, and there's a machine that does them, and so we kind of know what they are. Medications are easy because we make them. We know what we put in them. But things like diseases are a little tough. So what's the location of diabetes? Is it the bloodstream where the blood sugar is elevated? Is it the islet cells of the pancreas which are not producing enough insulin? Is it insulin receptors that are not sensitive enough to the insulin? Is it the vascular basement membrane that's being altered by elevated glucose? Is it glycosylated proteins in the vascular basement membrane? So what is the site of diabetes? It becomes tough to model things sometimes when you're dealing with higher level concepts. So what is biomedical informatics? The easy answer if I'm at a cocktail party and people go, informatics, what's that? I go, it's like computers in medicine. They go, okay, and then they move on to talk to somebody more interesting. But this is a definition that was published in JAMA a while back and gets often quoted. The field concerns itself with cognitive information processing, communication tasks of medical practice, education, and research, including information science and the technology to support these tasks. So it's a pretty broad definition, but it's really just a hodgepodge of other things. And in a sense, informatics, the field of informatics, kind of grew up that way as a hodgepodge of interdisciplinary things. You got a computer scientist and a doctor working together, or you got a guy with a computer science degree who got an MD and starts bringing these things together, or people with a cognitive science degree or psychology degree, and then they become nurses, or any kind of other sort of cross-fertilization between different domains. This is my definition, and of course I like it the best, but I think it's a little more inclusive. Art and science of organizing knowledge of human health and disease and making it useful for problem solving. So it's about knowledge, the knowledge that tomatoes are fruit, and making it useful for problem solving, what to put in a fruit salad. But it's all about solving some kind of problem. Is it a research question? Is it a patient care question? Is it a health policy question? Those are all problems, and so, as I say, very inclusive, but that's the end goal, is to answer some kind of question or solve a problem. Okay, so here, I don't know how well you can see that, we've got some DNA, it's a proteins and a Krebs cycle, and so there's this level down at the molecular level where we have molecular biology and genetics, and bioinformatics typically deals with things at that level. So it's sequence analysis. In fact, the term bioinformatics is a term that was used to talk about the informatics of not biology, but biotechnology. Biotechnology, you don't hear that word very much, but 20 years ago, the term biotechnology was used to talk about the process of sequencing biomolecular sequencing. So proteins, DNA, RNA, they called that biotechnology. It was a pretty general term for a very specific thing, but that caught on for a while, and so bioinformatics came from that. Then we get up to sort of the cellular level, and now we're getting into sort of translational informatics, where things are going from the molecular level to a functional level. And then we get up to the human level, and we talk about clinical research informatics. So we start looking at, okay, now we're going to run a clinical trial. What's the informatics that relates to that? Some of it could be modeling what's going on, but other things could be sort of administrative, like how do we keep track of who signed what consent? And how do we find people in a database to recruit them for our study? So it can be a lot of different things. And then we get patient care, and that's the health or clinical informatics, health informatics, and then population health, population informatics. Health informatics, probably the upper levels of this thing, things where we're dealing with human beings. So health and disease. Any questions about that? Anybody want to challenge me on any of those terms? Because people have lots of different definitions, and in fact, you'll see bioinformatics used as the general term for everything. And it can be confusing because people come to me and go, hey, I want to learn about bioinformatics. I go, well, I don't actually know a lot about bioinformatics the way I define it, but I could tell you sort of the spectrum, or I could tell you other parts like electronic health records, and they'll go, oh yeah, that's what I mean by bioinformatics. Okay, well, first we're going to talk about terminology, and then we can talk about what I know about it. So, questions about that? All right, so along with that spectrum of representation, of knowledge representation, then there's a spectrum of problem solving. So we can do things at the scientific base of biology and medicine. Think about the basic sciences, things that we're looking at first principles. Then we, in patient care and healthcare, we collect signals, very broadly speaking. Some of it is information that the patient tells us, some of it is laboratory results, some of it is EKGs, X-rays. We think of all that as kind of signals that we then interpret into some conceptual level. So an X-ray, a chest X-ray, nowadays it's digital and it's a bunch of pixels, right? But we don't say, well, the pixel here is white and this one's gray and this one's black. And we don't even say there's a collection of pixels here that's this size. We take it to the next level. We say, oh, there's an infiltrate in the lung. Or we say the lungs are clear. We say the heart's enlarged. So we talk about conceptual things that a computer wouldn't just be able to spit that out and say, oh yeah, here's the part where it's the heart and measure it. We have to do some kind of interpretation of the signal to get to the conceptual level. But yet we need to do that to talk to other people about it, whether it's the patient or other clinicians. We don't want to say, well, there was this big white area in this part of the X-ray, and have everybody go, oh, that means it's in the lung and it must be this. We go, no, there's an infiltrate in the lung, and so we deal with it at this conceptual level. And then we want to apply science and data to the process of clinical care. So now we've got our signals interpreted as concepts, and then we figured out, what do we do about these concepts? Which of these things are problems? Which of these are normal? Which are the ones we can do something about? And if we're going to do something, what are our options? And then we can extend that out to populations. So we can say, well, we've done a study of vaccinations, and we found that giving this vaccine prevents this disease. So now we're going to build something that works in the health system for individual patients. Oh, Mrs. Jones, you're at risk for this disease. We recommend giving you this vaccine. We can order the vaccine and so on. But then we want to say, okay, now how do we do this to populations? The questions become different. It's not just about an individual patient. Now it's sort of, well, how do we deploy vaccine centers? So when they were worried about smallpox outbreaks at one point, they said, well, how would we deploy a vaccine on an emergency basis if somebody weaponized smallpox and there was a big outbreak somewhere? You can't just, a lot of people have been vaccinated, older people, but younger people have not, and you want to vaccinate the population as quickly as possible. You can't just say to everybody, go to your doctor and get vaccinated, because a lot of people aren't close to their doctor, or their doctor won't have the vaccine. So if you wanted to deploy a vaccine center somewhere, how would you do that for the population that would maximize the number of people you could reach in a minimum amount of time, all that sort of thing. So those are different questions than you would ask at or try to solve at the patient level. All right, so I have a little case, and I just put it up there, a medical case, not too technical, but maybe make you think about some of the things, or we can talk about some of the things that we would use informatics for in healthcare. So, 50-year-old Native American female presents the emergency room with chief complaint of lip numbness, nausea, and chest pain. So that's not an unusual sort of chief complaint. I mean, it doesn't happen a lot, that particular one maybe doesn't come up much, but it's got all the same elements. We've got sort of a gender, we've got a racial element, we've got a chief complaint. That's a constellation of symptoms that we might look at that and go, oh, I know what this is, or gee, that's an odd combination. The patient was generally well, so now we have what we call past medical history of present illness. The patient was generally well until about a half an hour prior to arrival in the emergency room, eating dinner at a seafood restaurant at Rock Harbor, Massachusetts, finishing dinner of New England clam chowder, lobster, steamed clams, corn on the cob, when she noted onset of symptoms. Others in her party ate fish and chips, although 2 other people ate the clam chowder, none ate the steamers. So now you go, oh, maybe I'm thinking about foodborne illness, and maybe the steamers are at fault. So what kind of things do you get from steamed clams? So you might already think that. Well, how many people know what kind of illness you get from steamed clams? Nobody? What your computers know, right? So it would be easy to do a Google search to say foodborne illness, steamed clams, and you'd probably get a bunch of things back. But even better if you had a database or a knowledge base that actually had those answers already, there for you, so you could put in steamers and it would give you back, whatever list of diseases to look for and tell you what the symptoms, or even better, put in steamers, put in the symptoms in some controlled terminology and have it just give you and say, this is the most likely foodborne illness given that constellation of symptoms. So you could build a system that did that, but you're representing knowledge at some level that the computer can manipulate, right? Controlled terms, the relationship between symptoms and diseases, the relationship between diseases and foods. So we're representing things. It's not just a big jumble of terms, although that could work, and Google does that all the time, and it can be effective. If you want to be even more effective, you build something very specific. She gives a history of hypertension, states she's getting a capsule, half green, half blue, from her private doctor. Also reports treated for the past for tuberculosis while she was pregnant. Doesn't remember what she was treated with or for how long. She reports that another hospital on the other side of town had a liver biopsy, and she thinks the diagnosis was hemochromatosis. She has an allergy to buffering. All right, so now this is our history. This is our past medical history kind of thing. She's on a medication of some sort. Be nice to know what that was. She's got some kind of diagnosis from a liver biopsy. Be nice to know what that is. She's treated with, in the past for tuberculosis, and that's relevant not because she might have tuberculosis right now, but if we treat her with certain treatments we might give her might make latent tuberculosis more of an issue if we suppress our immune system, which we're about to do. Okay, and here's the last part. Physical examination, or second part. Physical examination revealed well-developed, well-nourished, diaphoretic, meaning sweaty female, moderate respiratory distress. Vital signs show pulse of 110, respiratory rate of 8, oral temperature of 100.3, blood pressure of 150 over 100. Examination revealed RALs over both lung fields. Abdominal exam revealed tender, palpable liver edge. Neurologic exam reveals dysarthria, diffuse muscle weakness, hyperreflexia. Okay, so we don't have to go into the medical aspects of what's going on here, but you can see there's some numbers that we've collected. There's some physical findings that we've turned into concepts. Like we didn't just say we feel something funny here. We said, oh, that's her liver edge. And we hear a bunch of sounds. We don't just record those sounds and play them back later and go, here's what the patient's lung sounds like. We actually interpret that as a kind of sound that we can then say what it is and where it is. Laboratory tests, so typical. We got blood sugar, from a serum one and a plasma one. We have a complete blood count, a bunch of numbers, finger stick blood sugar. So we see actually 3 different kinds of blood sugar here: serum, plasma, and finger stick. And the question is whether how those relate to each other. And another glucose test, but this time in the urine. And then we have a culture positive for a particular bacteria. And then we have a cardiogram, which is a signal, right? But we interpret that into, say, oh, that signal matches sinus rhythm at a certain rate and some other abnormalities that we might notice. Okay, the last part. So now that was physical exam, other objective data that we collect about the patient. We admit the patient to the hospital with a preliminary diagnosis, and now things start to happen to the patient, and that's the hospital course. Patient's nurse reported the patient was more worried about who would care for her elderly father if anything happened to her. So this is completely separate from her problem, but now how do we keep that in account when we're trying to take care of the patient? Because that's going to have bearing on the medical decisions that we might make. A medical student reviewing the case wonders whether paralytic shellfish poisoning, which by the way is the ultimate diagnosis in this patient, could cause myocardial infarction, which there was evidence of on the EKG. She decides to do a literature search to see if there's any correlation. So now we're bringing a computer system into play, PubMed, say, or Google, and now how do we bring that to bear on this problem? And the patient's treated with activated charcoal, stomach lavage, followed by enteric-coated aspirin. And then she has worsening respiratory insufficiency. She is intubated, placed on mechanical ventilation because we gave her enteric-coated aspirin to somebody who has a bufferin allergy, and they both have aspirin. And so maybe she's allergic to aspirin, and we gave her enteric-coated aspirin. Although the names of the drugs are completely different, but if you knew about the ingredients, you might say, wait a minute, this patient may be allergic to aspirin. All right, so we have data types now. Sort of at the lowest level. We have things we collect directly from the patient. We turn those into text that we write in our notes. We have numeric things that we collect, like blood pressure and laboratory tests. And so we have these quantitative things. And then we've also got the signals like the EKG and an image. And we interpret those and again turn them into some text kind of data. And then we try to the best we can now, code the data using some controlled terminology of laboratory tests or physical findings or diagnoses, that sort of thing. And we do that manually typically, but if we have structured text, sometimes we can use natural language processing to get into a coded form. And by coded, I just mean using a controlled terminology where each one has some code, 1, 2, 3, 4, ICD-9 codes or ICD-10 codes, but something that the computer then can say, oh, okay, here's a list of things that I can match up with things that I know in medicine. And then ideally, we want to get it to standard coded terminologies because then we can share data, we can share knowledge sources that have been created elsewhere that use standard terms and so on. So these are blobs, and they're just bunches of data. And the computer can store it and can give it back to us, but doesn't really do any what I call symbolic manipulation. It won't, it just says, oh, you want to see the x-ray? Here it is. You want to see the EKG? Here it is. Here's a list of what people said. Here are the numbers. And it'll just spit them back. These things, though, are symbols. They're things the computer can reason about and say, oh, well, this laboratory test indicates this disease, and the treatment for this disease is this medication. It can start to help us do things that are more sophisticated than just store the data, only show it to people who are supposed to see the data, and then show it to them when they need it, or when they request it. So now let's think about some questions that would be really good to answer in this patient and think about maybe a little bit about what kind of representation would be needed to do that. So how do we find out what the patient's medication's on? This is actually pretty important. A blue-green capsule. What is this? Is it a vitamin, or is it, is it a chemotherapy agent, or is it something for tuberculosis? It makes a big difference. How do we get records from another institution? That liver biopsy would be nice to know. Was it liver cancer? Was it really hemochromatosis? What's going on with that? And we have to make sure we get the records in a way that we can incorporate them into the records we have, so it's not just a big pile of paper or scanned in paper, but actually something that we can index and query against. And we have to make sure, of course, that we got the right person. We don't want the wrong records mingled in there. So we get the right records. And then we have to keep the clinicians from getting overwhelmed. We start bringing in all this stuff and go, here's all the data, good luck. And we do that typically sometimes. In the old days, the paper records, you plop that down and say, okay, doc, here's the patient. Make sure you don't miss anything, because if you do, we'll take you to court and sue you for not doing the right thing. What about our potential allergy, the buffer and aspirin allergy? How do we do that? How do we get relevant evidence from the medical literature that helps us figure out what to do? How do we bring expert systems in there that might give us a differential diagnosis for the symptoms that we saw? And then what about genetic predispositions that she might have? Maybe she's got hemochromatosis because she has some genetic abnormality. Maybe the drugs that we're going to choose would be metabolized differently in her based on her genetic makeup. That gets pretty complicated. How do we bring that into it? And then what do we report to public health? This woman has a foodborne illness. Do we have to report it, and how do we do that? Typically we'll teach physicians, there are diseases you have to report to the health department. If it's a medical student, say, okay, what are they? And go, well, it depends where you're practicing. So wherever you are, just make sure you go look that up and then remember that, and then make sure you do the reporting. And it changes from place to place. And then how do we take all that knowledge that we've gathered about that patient and generate new knowledge about, okay, what are the presenting symptoms of paralytic shellfish poisoning? Maybe, how often does myocardial infarction occur in this? We could use that information then to treat the next patient along the way. So all of this has to do with how we're going to represent knowledge, information about the patient, and knowledge in general that we can match up to that information. All right, so this is clinical translational science, right? So you guys are doing research. Some of you, hopefully, are doing research on human beings, and you might be interested in patient data, not just the data that you collect in a case report form or that comes back in a laboratory test that you've performed as part of your study, but you might also want to get other data about the patient. And the electronic health record is a place that people now are finding as a useful source of data. So why is it useful? It's captured by clinicians and instruments. So it's not just the patient filling out a form. Patients can tell us a lot accurately, but there's a lot of things they can't tell us accurately. They're not real good at physical exams on themselves, for instance, or even history taking. They'll be general. We can go to a past clinician's note about what was going on with the patient. And so the clinicians are a more objective source, hopefully a more reproducible, reliable, accurate source, although of course human beings make mistakes. But for instance, when we go and examine somebody's heart, the notes that we'll take on, say, describing the heart murmur will be pretty consistent, because we teach everybody the same method and some objective ways to describe it. And then of course we have instruments which are only as good as their accuracy, but they're pretty good. They have data about the patient's phenome. So you can go and do the genome that Bruce was talking about, but what does the patient look like? He was mentioning, recreate a phenome in a zebrafish. I'm like, oh, really? So a zebrafish has a myocardial infarction, or how does that work? I don't know how many phenomes you can, what color hair does a zebrafish have, and what gene controls that? I don't know a lot about zebrafish, but I'm pretty sure you can't recreate a lot of the things that you see in human beings because they're too complex. So electronic health records is a good place to get that. They're becoming universally available. 10 years ago, there was maybe 10% of hospitals had electronic health records, maybe 11%, something like that in the US. Big ones, of course, more likely, but small ones had paper records. Today, it's more like 85%. So that's because of the Affordable Care Act and the incentives that it gave hospitals and physicians for purchasing or installing these systems. And so it's much more available now, and as a result, there's more data that's at least potentially available, almost free. So it's not really free, but we treat it as if it's free because somebody's already collected it and just sitting in a database. And so, we have to do some effort to get the data out. We've got to hire a programmer, we've got to build a system. But once we do that, then we get those data flowing, and it's actually pretty inexpensive to keep up with those data. And then we can do post-market surveillance. So phase 4 studies are now a lot easier to do when we have all these electronic data. So phase 4 study is, phase 1 through 3, those are the research before you're allowed to market the drug. Once you market the drug, you want to make sure that the drug isn't causing problems in a general population, maybe a rare side effect that in the studies of 100, 200, 1,000 patients you weren't seeing it, or at least seeing it at a statistically significant level. But then when you go to a larger population, suddenly you see, oh, here's a thing that we didn't expect to see, but look at that correlation. It's undeniable. So we can do that relatively easily from electronic health records if we have good data. And you can use it to validate research studies. So you might say, gee, we think from this research study that when we treat people with this disease with this drug, 50% will improve. So that's your study of 100 people or 1,000 people, and you publish it, and then they market the drug. Now you can go to a larger database and go, is that true? Is that really happening? Because maybe there was some strange way we were doing our study that, we also happened to be making sure patients were getting good nutrition while they were in the study, but in the real world, they aren't getting good nutrition, and it affects the outcome of the study, of the outcome of the use of the drug. So we can validate that, and we can actually replicate research studies using electronic health records by, for instance, saying, well, here's 2 drugs. They're both approved for use in this disease. People choose them arbitrarily. So let's have a study where when somebody comes in and the physician's thinking of treating somebody for this condition, we offer to say, hey, how about enrolling this patient in this study where we'll let the computer randomly assign one of these drugs or another. And then we can follow those patients prospectively and see what happens and see if they, one seems to perform better or, another. It's called a pragmatic clinical trial. And that's being done quite a bit now because it's a lot cheaper to simply add that little selection process and then just go about normal course of care. So it all sounds really rosy, and maybe we don't have to actually do any research anymore. We just pull data and analyze it. There are some limitations of these data, and it's important to understand what they are. And that's where the informatics comes in. I'll talk about that in a little bit, but first I want to talk about I2B2. How many people know about I2B2? Wow, good. Okay, how many people knew about the $1,000 I2B2 contest? Only a couple people. So we actually had a contest. All you had to do was go use I2B2 and write a 1-page abstract on your findings. And we had a contest, gave away $1,000 first prize, 2 $500 second prizes. We'll probably do that again. It was pretty successful, so keep your eyes open. It's pretty, it'll take you 5 minutes to do the search and an hour to write up the abstract. Not bad, for $1,000. So keep your eyes open for that. So what is I2B2? So we have an electronic health record. It's got a lot of different names. Cerner, some people call it Cerner, but that's just the company that makes it. Some people call it Millennium. Millennium is the platform that Cerner creates. Some people call it a PowerChart, which is the name of their product. And some people call it Impact, which is our name for their product. So whatever, you'll hear these different things. But it's a Cerner commercial electronic health record system. And we extract data from that nightly, and we put it into an enterprise data warehouse, and we have analysts that can go and query that and answer questions. And that's great, because you can talk to the analysts, and they will go and get your data. Now, the typical way that happens is you go to the analyst and say, I have a question, and they say, we'll get back to you. And a couple weeks later, they go, remember that question you had? You still have it? Because we have time now. So you tell them the question, and they go off and they get some time to work on it, and they come back a couple weeks later, and they go, here's the data, and you go, that's not what I want. You go, that's what you said. You go, no, I wanted it this way, or oh, now I notice I should have gotten this other thing. They go back and they get it, they come back, yeah, it'd be really good if we got this extra thing. So you go back and forth, and that's pretty typical. Not just anywhere you're using some sort of secondary expert. But we now have I2B2, which is a self-service version of the data in the electronic enterprise data warehouse. It's more limited. At least right now, we're trying to move more and more data in there, but the idea is researchers can go and be their own analyst. And that has some advantages and disadvantages. Disadvantages are, you're not as familiar with what's in there. There's a learning curve. So we have classes to teach people, and we have contests to encourage people to try it out. And gradually, people are learning how to use this. The advantage is that it's self-service. You can go in there and do your query, and a few seconds later, get your answer back. And if you didn't ask for the right thing, nobody knows. You just fix it and get it, and eventually you get your data back. So you're not waiting for this, sort of human communication cycle to take effect. You just be your own analyst. And if you get a certain set of data and you say, hey, this is great, but now I need more data on these patients, then you can go to the analyst and get more data. And so this, of course, is all with the proper IRB approvals. If you're just getting summary data, it's considered not human subjects research data, and you can get that from I2B2 very easily. If you want a limited data set, which is sort of de-identified, we would think of it as de-identified, but it'll have dates in it, and officially those are considered identifiers or something you could use to identify a patient. But we think of it as de-identified. You can get those as well. They are considered exempt from IRB oversight, and we automate that process for you. So you don't have to submit a form, wait for the approval, and then go to the analyst. You just fill out the form online, and you get your approval right away, and you get your data. If you want a fully identified data, that requires approval from the IRB, either with a waiver of consent or requiring consent, whatever whatever they think is appropriate. And then then typically right now, the analysts will get those data for you if they're identified for. We also have connections to networks. One of them is called Shrine, another is called Trinetics, and they link us out to other sites that are also running I2B2. Basically, you issue your I2B2 query, and instead of getting back one set from UAB, you get back sets from all the different places that are on the network. Another one is Trinetics, which uses a different user interface, but also goes to the same databases. Okay, so what would you use this for? So this is electronic health record data, primarily, and it's right there, but now you're not querying it patient by patient by patient, which is the way you use the electronic health record. Now you're querying across patients. So what can you do with this? And these different boxes here are meant to be different parts of the cycle of biomedical research. And so you might say, gee, I'm interested in, studying patients with a certain condition. I wonder how many patients we have that have that. So you could use I2B2 or the warehouse to find out, get an estimate. Well, we've had 100 people last year, and you go, good, I only need 10, so that seems pretty good. Yeah?
One of the issues, I've done that, but for some rare conditions, and if you're looking to see how many people you have, because you cannot have access to the actual patient until you have IRB approval, you're sort of limited in what exactly you can get out of that.
Well, so you're limited, although with IRB approval. So for instance, if you're doing this sort of preparatory to research, you can go to the IRB and say, listen, I'm trying to do this feasibility assessment. I need to know how many people there are. I don't need to know who they are. I'm not going to contact them initially. I want to do this feasibility. That you can have that approval because the analyst will get the data for you. So you're not going to see identified data. So the analyst can get those data for you and just strip and give you a number. So that summary data can help. Now, if you can't get it through I2B2, there's a few reasons why that might happen. One is that it's a rare condition and we don't have a code for it, so we have some ICD-9 or ICD-10 code like, rare infection not elsewhere classified or something, and so it's no help. You'll say, well, it's somewhere in this group, but I don't know if it's all of them or none of them. You can't tell.
I was wondering if there's like an expedited IRB mechanism where we could actually go into the patient's charts,
yes. So yes,
it's go through the whole process of.
Yes. So you can either get, depending on the level of information you want, you can either get an exemption or IRB approval. The exemption is, is a paper form. You got to fill it out. You get it back. Then you can go to the analyst and say, I have an exemption. I need you to go and find all the patients that have that and tell me a number or tell me specific data, but don't give me any identifiers. And then they can do that. So a human being's looking at another human being's record, but it's not you looking at their record. It's an analyst. And so the IRB permits that.
But there's no way for me to actually do that.
So, all right. So first of all, if you became a warehouse user, which you could get training.
Which I am, but it doesn't give any identifiers. Like we have to go.
You would have to go to the IRB and get, but then they would likely give you a waiver, right? They'd say, because you're not contacting the patients, you're just looking at their records. And so they would say it would be impractical to get a consent. There's unlikely to be harm to the patients and so on. So they would likely give you, I don't speak for them, but that's typically how it works. And then you could go and do that. What we're trying to do is, move the text reports into I2B2. So you'll be able to do that through I2B2, which have you used I2B2?
Yes.
Have you used the warehouse?
Yes.
So I haven't used the warehouse. Which interface do you like better?
Just
Sorry?
where you query by, is that a new feature? I had the training that was last year where you entered, the ICV or whatever.
To use the warehouse?
Yeah. But maybe it's not what you're saying.
So is it I2? So there's a form you fill out, and a human being goes and searches the warehouse. So if
No,
you're
but
aware.
you can also have a login and do the search yourself, right?
Yes.
And it'll give you numbers of how many people meet that criteria.
Right.
I've done that. That's
Yeah.
ITB2, yeah.
Yeah, that's, well, let me see if I have a picture. And you tell me, does it look like that? That's I2B2. Okay, so there's also a warehouse, and the user interface is not as nice as that, but you can become trained on that. All right, but we want to move free text in there so that you'll be able to get at those. So if somebody's got a rare disease, hopefully somebody said it in a text note somewhere, and we can get at that. All right, what else do we have? So we want to pull data from populations. We could go out to Shrine or our own population, and we could use that information, for instance, to help us write a proposal as preliminary data. Then we can get this limited data set, an aggregate data, so we can actually pull the data and analyze it to answer a question. How often do people with this have that? And we could, even see that, or how's that doing over time? We can pull those data and answer some limited questions, which was the purpose of the contest. Then we can also use that to figure out, okay, who's really eligible for a study, because we want to contact them and enroll them in a study. And there, of course, you do need IRB approval. You can go look up the data to find the patients yourself if I2B2 has what you need. I think of the recruitment or enrollment criteria as falling into three categories. One is what I call easy, which is a thing like finding all the people that are taking this drug or have never taken this drug or something like that. Something you could find from I2B2 just by doing a query. Then there's hard. And those are things that are in the record, but they would be hard to find in an automated way. Somebody's got to read the record and kind of figure it out. So it's, patients who took this drug but didn't tolerate it. And that might be a reason you want to recruit them to give them a different form of the drug or something like that. Well, a lot of times you'll see, well, they were on the drug, then they're off the drug, and there's no easy way to tell why they were taken off, so you'd have to read the record. So the data's there, but it's hard. And then there's impossible. Impossible, and by that I don't mean you can't get the data, you can't get it from the EHR, because it's stuff like, are you willing to stop your drugs for 2 weeks while you're on the study? Well, that's not going to be in the record. Or you have to have had chest pain for less than half an hour. Well, if it's in the if it's in the EHR, by the time you've seen it, it's been going on a lot longer than half an hour. So those are things you have to do at the moment you see the patient when you're enrolling them. And I'm trying to figure out how do we create a mechanism for letting researchers define their criteria kind of at these three stages to make it easier for them to define their study, to do their initial cohort estimations, and also to communicate with analysts about what kinds of data they want to get. And if you can imagine that if we wrote, well, what's the easy criteria, what are the hard criteria, and what are the impossible ones, and then you would write up your study protocol with that in mind, it would speak directly to the data collection process that you use. And that's informatics research. Let's see, approved data research. So once you have your study approved, you can pull data on the people in your study, and it would be an identified data set, because now they're your subjects, either waiver of consent or maybe you even consented them to be in your study, and you want to be able to get your data from the warehouse. We're moving to being able to do that in I2B2, so that you could say, get me all the lab tests on everybody in my study. And now you hit one button and you get a spreadsheet that has all your lab tests. Instead of having to go to an analyst, have them set up a query, wait for the results, then go back to them and say, could you rerun it because I have more data? Or the way a lot of people do it, they go to the EHR, the Cerner system, and they log in, they look up a patient, they look at the lab, they write it down, they go to the next patient, they write it down. Eventually they've written everything down they want, then they go and they type it into a spreadsheet, and then they load that into something, and hopefully there were no transcription errors. Really tedious, sort of hard to keep up to date. So now you'll be able to do it when we introduce this in I2B2, you'll be able to just simply get the data right out in identified form. And then what's this? Additional data for participants. Not sure what that's in there for. And then you can go outside. For instance, if you want to do a multi-site study and you want to figure out what are the other sites that have patients that you're interested in, we can go to one of the networks and find out, oh, University of Kentucky has 100 people. Let me find a researcher there who's interested in collaborating on this. And I think that's it. Okay, so what's an I2B2? Hard to read back there, but it's the typical things, and these numbers are a little old, but they give you a sense of the volume. So let's see, we've got blood pressure measurements, 48 million blood pressure measurements, 36 million inpatient medications, 141 million laboratory results. So there's, that's big data. you could do some interesting things with things like that. And then fact counts by year, yeah, this is an old table. We got to get this up to date, but it's around something like 50 million facts a year are being brought into the system. So it's pretty big. All right, so this is the screen for I2B2, and over here is the terminology, and there are different ways to find the terms of interest. I typed in the word proton pump, and it found a class of proton pump inhibitors, and there's 7 things under there. And then I created a query where I said, find me all the patients with gastroesophageal reflux disease. That was from a previous thing, and just drag it over. And then I dragged over 3 of these terms, and I ran my query, and I got 27,000 patients who have had that disease and are on that drug. Now, it doesn't mean they're on that drug for that disease. It doesn't mean they had it at the same time. It just means somewhere in the record is one of these 3 drugs and something in that class of diagnoses. Now, here, I've created a class-based query. So instead of 3 drugs, I dragged over proton pump inhibitors, and now I have 30,000 patients because I didn't have to drag over the other 4. I just dragged over the whole class. It makes it a lot easier. You can change the parameters on your query. So if, say, you're looking up a medication, you can say what strength of the medication you're interested in. This was let's see, this is BMI, body mass index. You could say I'm interested in the body mass index above a certain number, below a certain number, between a certain range. Same thing with laboratory tests. So you can set parameters on there. You can say the patient doesn't have this, or you could say the patient has 3 of these. That sort of thing. So there are various things you can do to figure this out. Usually, though, you don't want to do your whole query this way because it'll be too awkward to create the whole query. Generally, what you do, create a query, download your data, and then you can refine it on the data that you've downloaded. So, for instance, if you said, find all the patients that had a proton pump inhibitor within 2 days of the diagnosis of, gastroesophageal reflux disease, it won't be able to do that very well. So instead, just download it and then look at the dates of these things and do your analysis that way.
So it's the medication that's not current. It's not like always, the system is not always updating. So for instance, the PPI or proton pump inhibitor, the patients who are taking a PPI today, would that be?
So not today, but and maybe not exactly yesterday, but maybe a week ago. So right now the warehouse is updated overnight, and then I2B2 is updated from the warehouse. And there's a lag. We don't do it every day.
So if the physician takes the medication off, that comes off of it?
No. So what's recorded are events. And so the events are this was prescribed or this was administered. And so when a medication comes off, then there'll be a discontinuation order, and so you can see that. But all the orders are in there. So you can say, find me all the patients. It might be hard to say, find me the patients that are currently on this medicine. But you could say, find me all the patients that got a dose of this medicine in hospital within the last year or the last month or something like that. It's hard to tell when somebody's not on when a medication's been stopped. Sorry?
You can actually do a date range if you're looking at like how many patients were on for this many months, and it'll be able to tell you something like that.
Yeah, so yeah, specific range. You can't say find me all the people that got this drug for 3 months. You can say from January through March, and it'll do that. But if you said, I need somebody that's been exposed to this drug for 3 months, I don't care which 3 months, that's an example where you would want to just download all the data, see the starting date, the end date, subtract them, and see if it's greater than 90 days. And then, but you would do that and you can do that in Microsoft Excel, that kind of thing. Other questions?
And so all these data, if it's in the free text on the impact, that wouldn't show up.
Right. You wouldn't be able to find that through I2B2. You'd need an analyst to help you do that. But what this is useful, again, once you've done this, you've defined a patient population of interest. And so then you can go to the analyst and go, hey, I did this query. I found 30,000 patients that I'm interested in. Now, can you refine my query? Or can you give me additional data on these 30,000 people that I can't get through I2B2? And they'll say, sure, that's easy because you've already done the hard part. You figured out who those people were that you're interested in, and that makes it very easy for them to do the second step. Okay, so aggregate results, we saw a couple examples. Here's another one, 107 patients, and you can get more details about how many males, females, that sort of thing. We have 5 genders. We have male, female, unknown, of one kind and node of another and unspecified. That's a whole different discussion of how many genders there ought to be. But, is it 3, or male, female, and other? Or is it like Facebook where there's something like 52 genders that you can pick to describe yourself? 52. Go look it up. It's pretty interesting. If you want detailed data, so that was summary data. If you want the limited data set, once you've done your query and say, I want to get the limited data set, you'll fill out this form that basically asks for a title of your study, a description of your study, a few sentences about what you're doing. If you're going to share these data with somebody else, you've got to put in their Blazer ID, and if they haven't done the appropriate certification to be able to look at human subjects data, it won't let you. It'll say, no, you can't give it to that person. And so you take them off the list or tell them to do their study, do their training, and then you can get it. And what kind of data you want, and then so medications, labs, vital signs, etc. And then you get a spreadsheet, and it comes in different forms depending on what you're asking for. But it'll have a fake patient identifier, which is good for the whole data set, but they're not the patient's identifier. It's generated for the particular set. There's a concept code, start date, end date, whatever other attributes you might have about that kind of data. So caveats. So remember I said it's tricky to use EHR data because EHR data is not like data that you get in a clinical study where people say, okay, here's a form. Let's make sure we answer all these questions. And if you don't answer one, the system won't let you go forward. It'll say, no, you didn't answer this question. Please tell us if you're allergic to anything. Please tell us if your mother has any diseases, whatever it is. And so when you get through in a clinical study and you have a case report form and a counter form, you have complete data. So you want to avoid missing data. Electronic health records, not so easy. They're not created that way. They're created to take care of the patient, not to collect a complete set of everything that's known about the patient that might be reused for research. So with that in mind, you have to realize that the EHR may have inaccurate or incorrect data. So, for instance, a patient may think they have a disease, and so we put it in there, or we may think the patient has a disease, and so we put it in there, but in fact the patient doesn't have the disease. And an example I just was reading this case report of a patient who had, they thought had sarcoidosis, and they were treating, and for years and years, the patient wasn't getting better, it was getting worse, and then they finally figured out, oh no, it's not sarcoidosis, it's tuberculosis. Well, that's great, that patient, now getting appropriate treatment, but if you were to reuse that patient's data and look at the diagnoses, all those sarcoidosis diagnoses are still in there. They didn't go back and go, yeah, it wasn't really sarcoidosis. Turned out it was tuberculosis. The EHR still has the old data. So it may be inaccurate or incorrect because, it hasn't been updated. Patient may tell us something that isn't true, or a physician may incorrectly make a diagnosis and that ends up on the problem list. It's hard to get it off the problem list. EHR doesn't always tell us the complete story of the patient. The patient may actually go elsewhere for care, get put on different medications, have different tests done, and it may not all be in the EHR because patient's data may be spread out over many EHRs. Sometimes the data have been transformed for purposes other than researching patient care. So the ICD-9, ICD-10 codes are a classic example. So you take a perfectly good diagnosis and turn it into infection of the lung not elsewhere classified. And it's like, well, what do I do with that? Do I give them medication not elsewhere classified, or what do I do with that? And so you lose that level of detail that you might want for research. The data in the text form may not be recoverable in a way that you can use. So a lot of the data, like the example of the diagnoses, may be buried somewhere in the text, and you say, well, let me just look for that term. And you can try that, and sometimes you'll find it, but other times maybe the diagnosis is in slightly different ways. And a lot of times you'll get, it'll just say no evidence of whatever the thing is you're interested in. In fact, I was doing a project once where I said, I built this database. Somebody said, oh, I think that brain atrophy occurs in hypocalcemia. And I'm like, okay, well, let's go. We'll find all the patients with hypocalcemia. We'll find all the patients that have any kind of brain scan or x-ray, anything of the head that has the word atrophy in it. And we found like 30 patients out of 40,000 patients that had those two things. And every one of them said no atrophy, atrophy not present, no evidence of atrophy. None of them actually had atrophy. So it kind of refuted his hypothesis. But it was painful. Had it been 30,000 against a million patients, it would have been impossible. So the text notes may be tough to get the information you need. There may be multiple sources of data that end up in the record, and some may be more reliable than others. You might interpret things differently, so you need to know where did this come from? How did it get here, and how reliable it is. It isn't sufficient for it to just be on a problem list or appear in a report somewhere. And then the granularity of electronic health record data may not match what you need for your research. So a classic example is a pain scale. Okay, so I see a patient and I want to say the pain is better, or the pain's all gone, or the pain's worse today, because I'm looking at tracking that patient, whether she's getting better or not. But a researcher wants to know, well, is it a 0 or a 1 or a 2 or a 10 or a 20? And every study, of course, uses a different pain scale, and we don't, in clinical care, say, well, let's capture your pain on these 3 different scales in case anybody might need it someday for research. So we don't have it at that level of granularity that's needed. And then when we look at the course of a patient and compare that to what we would do in a clinical trial, they may be different, because in a clinical trial, we're often doing something different than the standard of care. That's the whole point of the trial, is to discover a new way to treat patients. And so we may not, for instance, we may say, okay, we're going to treat this patient and then do this test, and then we're going to do this drug or change this dose. And those things don't happen in patient care, either because we haven't determined that that's the right thing to do yet, or we don't test as often as you might test in a research study, because a research study wants, I need to know the blood sugar every day for the next 30 days, whereas in the medical side, we go, well, the blood sugar is normal, there's no reason to repeat it, and you won't have those data that you need. So lots of problems with this. So there are ways to get around this. We don't have time to go into those. We may be out of time now. How are we doing? 10:59. So let me just see if I had any really important things to say. So we'll get you these slides. Yeah, there's no way I have time to go through this and how all these different systems relate to each other through informatics, but that'd be a whole lecture, I guess. There's a lot of things that are making health informatics sort of mainstream now, so people have actually heard of this. Trying to reduce healthcare costs, reducing errors, functional genomics. There's the explosion of knowledge that we can bring to bear is making it more important to have health informatics systems. Mobile computing, other sensors, my Fitbit, putting my Fitbit data in there might be useful. And this whole interest now in big data, along with the emergence of electronic health records, the ubiquity of health records, suddenly we can start to study things in new ways. So there's lots of aspects of public, sort of contemporary science and politics that are driving this interest in health informatics, which is nice, because I've been in this field for 40 years and nobody paid any attention to me for the first 30. So now finally people are interested in this. So let's just see. We'll get you these studies. And if you're interested in health informatics, the American Medical Informatics Association is a good thing to check out. They have student rates. They have a conference every year, this year in San Francisco. Typically it's in November, typically in Washington. They move it around. And that's a good place to go if you're interested to see what's going on in that field and how it might apply. They also have other meetings. They have one on clinical informatics, one on translational informatics. So there's lots of different offerings. So just check them out, amia.org, see what they're up to. Okay, so I think that's my last slide. So it is very much the blind man and the elephant, and this is just scratching the surface, of course. But if you're interested, we're developing a master's program, a PhD program in the health in biomedical informatics, and there'll be both bioinformatics tracks and there'll be a clinical informatics track. So opportunity to get more exposure to that. We have some intro courses that will give you a broader view, open to everybody. So there's lots of opportunity to learn here. Okay, so that's the hour, and you probably have to rush off, and maybe there's another lecture, but I'll be happy to stick around and answer questions. Thanks.